Hello Nandish, how are you? Hello guys, how are you all? How is your studies going on? Okay, so good morning guys. Today we are going to start. From today onwards, I am the teacher who is going to be continue your classes. Okay, so my name is Manjuda. Okay guys, I am your biology teacher from today onwards. Okay guys. Okay, so guys, from today onwards, we are going to talk about the high weightage topics. Okay, so not like the chapter wise we are going. So it is the high weightage topic we are going to learn about. Okay, so this chapter I have taken for today. This chapter is cell. You know about the cell unit and cell cycle and cell division and the biomolecules. Okay, today and tomorrow we are going to complete this. Okay, cells. The important topics from the cell and cell cycle and division and the biomolecules. Okay, guys. And what is homework for you? You have to read and come these chapters. Okay. So, from this chapter, you are going to get 40 marks. Okay. From these three topics, you are going to get the 40 marks, students. Okay. So, from today and tomorrow, we will continue and complete these three topics. So, let us start the class. Okay. Fine. So, let us start the class. Before going to start the class, can you tell me whether I'm audible and screen is visible, guys? Okay, fine. Let us start. Yeah. So tell me in the technical poll whether I'm audible and screen is visible. So YouTube people from the chat box, you can tell me, guys. How was the Josh? How was the Josh, guys? You're preparing for the exams. Nandish? Yes. Very good. So let us start the class. Okay. So obviously the class starts with the question, guys. Too high. Very good. Okay. The question is here. Okay. The question is simple. I think everyone will answer. In the last class, we have discussed about this. Okay. Fine. So yeah, the question, guys. Which of the following cell organelle is responsible for extracting energy from carbohydrate to form ATP? Okay, I think everyone will answer. So guys, start answering in the YouTube also fast. Yeah. Yes, I got very good, Nandish. Yes. So student, this is a crash course. So you all know that we are going in a fast. Okay, if I was very fast means tell me in the chat box. I will little bit slow myself. Okay, fine. Yeah, very good, Nandish. Yes. Fine, I will not answer the poll. You can answer me in the chat box only. Yes, guys. So when we are talking about the extracting the energy, in the last class we have studied about the electron transport chain and the oxidative phosphorylation, right? Yes. So here the electron transport is linked with the oxidative phosphorylation, so that is the ATP synthesis that we have learned in the last class, right, guys? Yes. So that is the reason. So where this electron transport chain is going to be present, guys? In the inner mitochondrial membrane, right? Yes. So that is the reason option C is the correct answer, right? So now let you tell me, so why the option A and option B and option D is wrong, guys? Can you tell me? Why is all the not the correct answer? Nandish, can you try? YouTube people, can you try, guys? Ribosome. Let us talk about the ribosome. You all know that ribosome is a protein factory, right? It won't be possible. Why every work will be done by the only one person or only one organelle? There is a division of labor in the organelles in the cell, right? Yes. So ribosome is the protein synthesis. So that is a protein factory, right? So it is helps in the protein synthesis. So that's the reason that is wrong. Then coming to the chloroplast, even chloroplast has the main function, right? What is the function, guys? Food synthesis, right? In the plants, it is having the food synthesis, right? So that is also wrong. So then coming to the lysosome, very good, Nandish. Lysosome is a suicidal bag, right? If any protein want to suicide means it will go to the suicidal bag and it will suicide. Is that, guys? Is that going to be suicide itself in lysosome? For example, any bacteria enter in our body. So you know about the phagocytes, right? Our neutrophil and the macrophages. So that will engulf, that will eat the bacteria, and it will join the hands with lysosome, right? So then it is going to be broken down the bacteria into small pieces, right, guys? Right, guys? Yes. Lysosome contains so many numerous enzymes. It will break carbohydrate also, nucleic acid also, everything. We will learn today. Okay, fine. So it is the suicidal bag, boys, right? Suicidal bag. Right, guys? Okay. So this is it. That's it for the question. So here is another question. Select the mismatch. Okay, guys, you have to select the mismatch here. Carefully understand. Okay. Whenever the mismatch is here, only one is mismatch here. Okay, only one is mismatch. So you have to found that. Okay. So think, guys, think fast. Which is the correct answer? Very good, very good, very good. Plant cell has a big vacuole. Very good, Nandish. Yes. Yeah. Coming to the first option, guys. Okay, gas vacuole. You know about the gas vacuole, right? Where it is going to be present? Either gas vacuole is going to be present in us. In the humans, it is going to be present. No, right? We are going to, is we going to live in the water and are we going to float? No, right? The bacteria, whichever the bacteria which are present in the water, so that is helps in the buoyancy, right? Gas vacuole is helps in the buoyancy, right? So that's why this gas vacuole is going to be helpful. Yes, Nandish. Gas vacuole is correct answer, guys, right? Yes, correct. Then coming to the option C, protista, right, guys? So protista is eukaryotes only, right? When you talk about the fungi, protista, and plant kingdom and animal kingdom, these all the things will come under the eukaryotes, right? So that is also correct, guys, right? So that is also correct. Then coming to the option D, methanogens. You have studied about the gobar gas, right? Gobar gas, right? So what will happen? From the methanogens, methane gas is going to be produced, right? So is that methanogens is a eukaryote or prokaryote, guys? Bacteria, right? So now you tell me, guys, where it is going to be present, methanogens? Yes. No. That is not eukaryotes. That are prokaryotes, pa. Nandish, that is prokaryotes, not eukaryotes. Yes, correct. Gut of the ruminant, that is correct. But that is not going to, yes. Yeah, see here, methanogens, correct. What you are telling, that is correct. So that is present in the gut of ruminant. Gut of ruminant. Ruminant is nothing but you can take a cows, right? Ruminant. Okay. So it will helps in the breakdown of cellulose, guys. Okay. It will helps in the breakdown of cellulose. Okay. It is helps in the breakdown of cellulose, right? So then it is helps in the formation of methane gas, right? So that we use it as we will use the cow dung, right? So in that methanogens are there, it will produce the methane gas, right? So that's why we are using that, right? So that is also correct, right, guys? Correct, Nandish? Nandish, correct or not? YouTube people? Yes, Jennifer, correct, pa, correct. Jennifer, correct, yeah. Methanogens comes under the Monera, na? Yes, Monera is prokaryote or eukaryote? Tell me now. Now you tell me. Monera is prokaryote or eukaryote? Yes, prokaryote. Yes, guys, yes. So then coming to the option B, large central vacuole. Is that true, guys? When you talk about the animal cell, there is a negligible or very small vacuole is present, right? We know that vacuole is like a dustbin, right? Not like a dustbin. It will store the unwanted materials and water and minerals in it, right? So in the plants, it is in a very large. It is very large size, right? 90% it is made up of, right? Yes. When it comes to the animal cell, it is negligible or very small, right? So that is the reason option B is the correct answer. That is a mismatch here, right? That is a mismatch, right, guys? Yes. Fine. So I think you understood now, guys. Give me thumbs up. Give me thumbs up if you understood means Vibha, Surya, Shanta, Nandish. Give me thumbs up if you understood means. Jennifer, do you understood means? YouTube people, give me thumbs up in the chat box. Very good. So what about YouTube people? Give me fast. Yeah, no next question. Let us go to the topic. Okay, I will ask in between questions. Let us see how many of you will answer. Okay, guys. Okay. So you all know about the cell, right? What is a cell? For example, if you take a building, so for the building, how the brick is going to be a building block, like that, in our body, the cell is a building block, right? Each cell will combine. For example, so many cells will combine together to form the tissue, and tissue will form the organ, then whole organ system, then organism is going to be formed, right, guys? Correct or not? Correct or not? Very good. Yes, structural and functional unit, right? Yes. So now, when we are talking about the cell, there are two types, right? Pro means what? Before the cell was came, so that we will call it the prokaryote or the primitive cell, right? That is in the bacteria we can see. Then after that we have seen the eukaryotes, right there. So let us see what are the main difference between the prokaryotes and eukaryotes. Okay, fine. Let us come. You also have to answer me in the chat box. And yeah, what are the differences? You can also be, tell me in the chat box, guys. Okay, you two people, fine. First, let us come to the DNA. Okay, guys. So what is the DNA in the bacteria, guys? So that is naked, right? Tell me now, what do you mean by naked? Yes, Nandish, in building iron rods also use. Fine. You tell me, guys, now. Yeah, naked means what? Actually, what is there? Lack of nuclear membrane. No. Yes, here naked means DNA not having the protein. So that is a histone protein, right, guys? So that's why we are calling it a naked DNA. Okay, naked means in the eukaryote, the DNA is covering with the cloth. Cloth are like what we will call it as histone protein, right? But what happened in the prokaryote, it does not have histone protein. Remember, guys, okay? Does not have histone protein. When coming to the eukaryotes is having the histone protein. Okay, guys. Now you tell me, histone proteins are rich in which amino acid, guys? Which amino acid? I will tell you. When it comes to the nucleus, I will tell you, Nandish. Okay. Arginine and another amino acid. Tell me first. Another amino acid, guys. Arginine is correct. Histone is made up of the basic protein, amino acids only, we know that. Now you tell me, which is the amino acid that is basic in nature? Arginine and, remember guys, in the molecular biology, they will ask you this question. Arginine, right? Arginine and lysine are the amino acid. So the histone is very rich in this amino acid, arginine and lysine. Okay. So now let us come to the structure of the DNA. So how the structure of the DNA in the prokaryotes? It is like circular. Okay, it is a circular DNA. So obviously that is double membrane. So double only. Okay. So then coming to the eukaryotes. Here in the eukaryote, the DNA is linear, guys. Linear. So linear means what? Like this. Okay, linear. So here what will happen? It is a circular DNA. Okay, guys. So have you know about the two things, right? DNA is lack of histone and DNA is circular in prokaryotes. In eukaryotes, it is having the protein. So that is the histone protein is there and it is a linear. Right, guys? Okay. So now you tell me in the processing, I think you studied about the molecular biology, right? In the prokaryotes, it does not have any types of the, yes, Vibha. It does not have any type of the introns. Okay, guys. So what do you mean by introns? For example, if you take the eukaryote, so here we have a exons. And 1 minute. Yeah, I will take the another color. Okay. Yeah. So here it has here it is having the introns. Here you are having the exons. Okay. See here. So here what will happen? So here it is the intron. Okay, this is the intron and these two are the exons, right? So these introns are non-coding, right, guys? How many of you will tell it is non-coding? Studied long ago. Nonsense codon. No. Those are the non-nonsense codons. Not the nonsense codons. Removed by the process of splicing. Very good, Nandish. Yes. So whenever the splicing was happening or whenever the it will happen in only eukaryotes, guys. Okay. So whenever the DNA was, so whenever the RNA was maturing, introns will be spliced or introns will be removed. Okay. So then what will happen? So we will join the exons. Okay. These exons are the coding. These are the coding genes. Okay. So these introns will not code. That's why we don't want. So that's why we'll remove that. Right. Now, which are the coding regions are there? We will join that. Right, guys? Yes. So that is the introns, okay? That is not at all present in the eukaryote. That is not at all present in the prokaryote. Okay, guys? And have you seen? So in the prokaryotes, like the tailing and earthing is also done. They are nonsense codons. No, they are they are not the nonsense codons. Okay. So now let us come to the organelles. Okay. So now let us come to the organelles. Now you confirmed, right? Prokaryote does not have the introns, whereas the eukaryotes have the introns. So guys, have you remember when the introns are spliced, then we will add the tail and the capping, right? For the mRNA, capping is there and no introns are there, and then it is having the tail, then it is ready, mature mRNA, right? Then we will send that to the cytoplasma for the protein synthesis, right? Fine. So this is about the DNA, right? Okay, guys. If you have doubt, ask me. So now let us come to the nucleus. How the nucleus in the prokaryotes? Okay. So no nucleus only is there in the prokaryotes, right, guys? Because the DNA is suspended in the cytoplasma, right? It is not having nucleus only, or any organelle is not at all there in the prokaryotes, right, guys? So now you tell me which organelle is common in the eukaryotes and prokaryotes? Tell me, guys, which organelle is common in prokaryotes and eukaryotes? Think and tell me fast. Think and tell me fast, guys. Answer me in the chat box, YouTube people. Which organelle? YouTube people, answer me in the chat box. Very good, Nandish. Yes, ribosome, right? Is that having the membrane, guys? Is that ribosome having the membranes? Tell me. No, right? No. Ribosome does not have the membrane, right? So that's why we are telling that is common in the prokaryotes and eukaryotes, right, guys? Yes. So we don't have the nucleus in the prokaryotes, whereas nucleus is present in the eukaryotes, right? So no membrane bound organelles, nothing membrane bound organelles are present in prokaryotes. Then coming to the eukaryotes, membrane bound organelles are present. We are going to study about that. Okay. Very good, Nandish. In the coming slide, we are going to learn about ribosome. Okay, this is the very important guy. When it comes to the prokaryotes, we have 70S ribosome. Remember, guys, 70S ribosome. Coming to the all the eukaryotic cell, okay, except in the organelles like the in the chloroplast and in the mitochondria, we are having the 80S ribosome. Okay. When we are talking about the mitochondria and chloroplast, I will tell you which type of the ribosome is there. Okay, fine. So here, which type of the ribosome we are having, guys? 80S, right? So when you are talking about the 70S, it will be 50S plus 30S, right? So we are not going to add that, ma'am. When we add that, it will come 70S. No, sorry, 80S. No, why not 70S, ma'am? No, guys. So that is about the sedimentation constant. Okay. So that is not about the adding the 30 and the 50. Okay, guys. So that is the 30th subunit and the 50th subunit. Okay, remember 30th subunit and the 50th subunit. When we are talking about the 80th, it is consisting of 60th and the 40th subunit. Okay, so prokaryotes is having the 70th, eukaryotes are having the 80th. Recall. Okay, fine. Then how is the reproduction in the bacteria? They're simple, no? So just they will do the binary fission. That's it. Okay. So you know that bacteria are the immortal, right? In the living world, we have studied about bacteria are the immortal, right? They're not at all having the death, right? So the single parent will divide and it will give rise to the two daughter cells, right, guys? Yes. So it is by the binary fission, and here it is the mitosis and by the meiosis. Okay, fine. So let us come to the chromosome. It is having a single chromosome, whereas here having the paired chromosome. Then coming to the size, obviously there is a smaller in size, and here it is having the larger size, about the 10 to 100 micrometer. So whereas here in the prokaryotes 1 to 5 micrometers. Very good, Nandish. Yes. So do you understood this, guys? Do you understood this? Tell me yes or no if you understood this or not. YouTube people, answer me in the chat box, guys. Do you understood or not? What about ostrich egg? Yeah, those are the basic topics. No, whenever you study the chapter, you'll come to know that here we are covering only the main tough topics, guys. Okay. Yeah, you know that ostrich egg is a very large egg, right? Yeah. Which is the smallest cell, guys? Smallest cell? Smallest cell, can you tell me? Which is the smallest cell? Mycoplasma, right? PPL, we'll call it as the PPLO and the mycoplasma also. That is having the 0.3 micrometer. Right, guys? Smallest cell, mycoplasma. It is not having the cell wall also. Right, guys? True. Very good. Yes. Let us go to the next. Okay. Fine. Let us talk about the prokaryotic cell first. Okay, guys. So listen carefully. Okay. So we'll come from the outside to the inside. Okay. Outside to the inside we will go. So if you take, this is a bacterial cell. Okay, guys. So this is a bacterial cell. It is covered by 3 layers. Okay. We will call it as a cell envelope. Okay. So which are those? Okay. So first it is having the outer coat, guys. Okay. Outer coat is there. Outer coat. We will call it as glycogelase. Okay, we will call it as glycogelase. Okay, so outer coat we will call it as glycogelase. Okay, so then coming to the inside, after the glycogelase, which one is there? Can anyone guess? Can anyone guess, guys? After the glycogelase, what is there? Guess. First, yes, guys. Tell me in the chat box. After the glycogelase, what is there? Only Nandish is answering. What about other girls? What about other guys? Shanta, Surya, Vibha. This revision, no, guys. Tell me. No. It is having peptidoglycan layer, right? Glycogelase is there. Afterwards, peptidoglycan is there. Peptidoglycan. Okay, guys. Then inner. So afterward, what is there? Cell membrane, or we will call it as plasma membrane. Right, guys. So these 3 including, what are those? Glycogelase, peptidoglycan, cell membrane. These 3 together we will call it as cell envelope. Cell envelope. Okay, guys. Cell envelope is made up of cell membrane, glycogelase and peptidoglycan. Okay, guys. Fine. So this is the outside. First we will learn about the glycogelase. Then we will go to the peptidoglycan. Then we will go to the cell membrane. Okay, fine. See here, this is a bacterial cell. So here you can see the green coat, right? Green coat. So that is nothing but the glycogelase. Okay. Then you can see the white color between, right? White color. So that is a peptidoglycan layer. So then coming to the blue color is the plasma membrane. Okay, guys. Okay. So let us talk about the glycogelase. Okay. So what is the main function of this glycogelase? Why it requires the glycogelase? So this glycogelase usually made up of the carbohydrate. Okay, guys. Carbohydrate and some protein. Okay. And what it is actually doing? So it is the outermost covering of the bacteria. It is helping the bacteria. It is helping bacteria actually. Okay. It is helping the bacteria to escape from the host. Okay, escape from the host when it is engulfed, when it was in the endocyte, whenever it was in the host, whenever it was in the host, it will help the bacteria to escape from the host immune system. Okay, so it will not allow the neutrophil to recognize this bacteria. Okay, so like that, what it will do? It will do the protection. It will give the protection. And second thing is, it will protect the bacteria from the outer environment. Okay, whenever it is outside, so it will protect the bacteria from the outer environment. Okay, guys, two functions are there. One is protecting from the environment, and second one is protecting from the immune system of the host. Okay, immune system of the host. That is another function. Okay, so now let us go to there are 2 types, guys. Okay, so if it is very thin, glycogelase was very thin, so then we will call it a slimy layer. Slimy layer. For example, if you take blue-green algae, if you take the blue-green algae, so if you whenever in the it will like slimy layer it is having. Whenever we put a leg on that, it will be like what we'll call it slippery, right? It will cause the slippery like sensation, right? So that is what is called as a slimy layer. Okay, slimy layer. So that is thin. Okay, guys. So then if it is a thick, so that we will call it as a capsule. Okay. So remember, guys, if it is a thin, we will call it as a slimy layer. If it is a thick, we will call it as a capsule. So that is about the glycogelase. That's it, guys. What is the function? It will give the production. And there are the 2 types. One is the thin, the slimy layer, and second one is the capsule. Right, guys? Okay. So we have completed about the glycogelase. Do you understood this, guys? Vibha, Surya, Nandesh, Nagveni. Do you understood, pa? YouTube people, answer me in the chat box. Do you understood this? Yep. Okay. Fine. Give me a thumbs up. Thumbs up. That is enough for me. Okay. Fine. So now let us go to the peptidoglycan layer, guys. Okay. So our cell wall, not our cell wall, bacterial cell wall is made up of the peptidoglycan layer. Right, guys? 1 minute. Yeah. Thank you, Nagveni. So it is made up of the cell wall. So that is a peptidoglycan cell wall. Right, guys? So this peptidoglycan is also called as a murine. Okay. If it is in the multiple choice, they are given the murine means, ma'am, we didn't study that. What is that murine? No, guys. So that is a peptidoglycan. Okay. So peptidoglycan we also call as a murine. Recall, guys. Recall from the biomolecules. Peptidoglycan you have studied, that is the heteropolysaccharide. Right? Heteropolysaccharide. Right. When you have studied about the homopolysaccharide, you have studied about the stars, you have studied about the glycogen, where all the units is made up of the glucose. Right. But here what happened? One unit is NAG and one unit is NAM. One unit is NAG, one unit is NAM. Right, guys? So it is made up of N-acetyl-muric acid and the N-acetyl-muramic acid and the N-acetyl glucosamine. Okay. So what is the component of the peptidoglycan, right? Easily we will tell that NAG and NAM, right? What do you mean by NAG? N-acetyl glucosamine. And what do you mean by NAM? NAM means name. Now you tell me, NAG means NAG or name means NAM means name. Is that, guys? No, right. NAG means what? N-acetyl glucosamine. NAM means N-acetyl muramic acid. Okay. That is about the cell wall, guys. You know all about the gram, right? Gram staining, right? So based on the gram staining, we have divided this bacteria into two types, right, guys? Can you tell me which are those, guys? Can you tell me which are those bacteria? Now I want answer from others. Sushma, Vibha, Surya, tell me, guys. Which are those two types, guys? Tell me first. Which are those two types? Fast, answer me in the chat box. Very good, Melita. Very good, Pratiksha. Lakan. Good morning, Lakan. Yeah. Tell me fast, guys. Tell me in the chat box, whether in the YouTube or... Yeah. Very good, Vibha. Yes, ma. Gram positive and gram negative, right? So what we have to understood, the gram positive bacteria will retain the crystal violet, right, guys? So when we are talking about the gram positive, 1 minute, yeah. So when we are talking about the gram positive, it is having the thick peptidoglycan layer, right? Thick. It is having the very thick peptidoglycan layer, right, guys? And then you tell me which gram stain it is going to take. Crystal violet or safranin? Tell me, guys, whether it is taking the crystal violet or safranin. Recall. Gram positive looks purple or blue color, right? Now tell me. What about Nandish? Nandish became silent. Nandish. Well, which staining it will take, pa? Gram positive bacteria. Crystal violet, right? It will take crystal violet, right? For the gram negative bacteria, it is having thin peptidoglycan layer, right? So after we will use the mordant, right, guys? Iodine as a mordant we will use, right? So then we will wash, right? We will wash with the ethanol. So then again we will use another stain, that is a counter stain. That counter stain is nothing but safranin, right? So that we will use. So that is a gram negative bacteria will be stained, right, guys? Okay. So other than this, we are learn about this peptidoglycan layer. So we know that peptidoglycan is a protein layer, right? It is made up of two types. Obviously, you know that the amino acids are the L amino acids. When we are talking about the glucose, that is a D-glucose, right? Common, right, guys? That is common actually. Okay, if you are not studied also, I am telling. Usually the amino acid will be L. levorotatory. L in the chemistry you have studied, right? Levorotatory. Okay, common in our body also we are having the amino acid, right? So that are the L amino acid. Okay, but D amino acids are very rare, guys. Okay, so in this peptidoglycan, what happened? It is having both L amino acid and the D amino acid. So that's why what happened? Our protease enzyme can't break this peptidoglycan layer. Okay, so that is the reason. Okay, fine. So this gram-negative bacteria is consisting of the diamino-palmitic acid. Okay, diamino-palmitic acid in the cell wall. Remember, guys, gram-positive bacteria is having the diamino-palmitic acid in the cell wall. Okay. So then coming to the gram-negative, it is having the teichoic acid. Recall, guys. Remember. Okay. So it is having teichoic acid. So gram-negative bacteria. Okay. Fine. Sorry. The gram-negative bacteria is having the diamino-palmitic acid, whereas the gram-positive bacteria in the cell wall, it is having the teichoic acid. Recall here. Okay. Gram-positive bacteria is having the teichoic acid. In the multiple choice, they may ask you. Right. Like this question. The gram in the statement wise question, the gram-positive bacteria is having the teichoic acid. Correct or not, guys? Tell me. Gram-positive bacteria is having the teichoic acid. Correct or not? Give me thumbs up if it is correct. Correct. Very good. Why Lakan? Okay, 1 minute. 1 minute. Someone is asking question. Yeah, Lakan, I will try to share. Okay. Yeah. One minute. Yeah. Okay, I will try to share. Okay. Yes. Obviously correct. Very good. Yes, guys. Okay. Fine. So this is about the peptidoglycan, guys. Okay. Recall. What are the things you remember here about the peptidoglycan layer? Remember, it is a heteropolysaccharide, and it is also thick in the gram-positive and thin in the gram-negative bacteria, and it is made up of the NAG and NAM. Okay. So these are important things. Okay. So now let us go to the next. What is that, guys? Plasma membrane. That is the third layer, plasma membrane. In detail, we are going to study in the eukaryotes. Just I will give a hint here. Okay. So it is similar to the prokaryote. It is similar in the both prokaryotes and the eukaryotes. Right, guys? Same. What is there in the plasma membrane? Plasma membrane is consisting of the phospholipid. And in between, there are the proteins are there. Right, guys? Detail we will study today. Okay. So now there is a unique structure is there in the prokaryote. Unique structure. What is that? Mesosome, guys. Mesosome. What is this mesosome actually? See here. So this is a plasma membrane, right? This is a plasma membrane. What will happen? This plasma membrane will form the invagination. Okay, invagination. How it will form? It will form the invagination like this. Okay, guys. So why this invagination is helping? I will tell you. Just recall that, remember that. What is the mesosome? Mesosome is going to present in only prokaryotes. Okay. So these are infolding or invagination of plasma membrane. That's it. Then what are the uses of this? Remember, guys. So these are present in the bacteria. In bacteria, we know that it is not having any type of mitochondria, right? From then where the electron transport chain is going to be taken place, or where the ATP is going to be synthesized, guys, in the mesosome. Okay, in the mesosome it is happening. Okay, so let us see the functions of the mesosome. It increases the surface area. Okay, it increases the surface area. For example, if you see in the cupboard, if you take the cupboard where you will keep the cloth, are you going to keep the cloth like the however you want? If you keep however you want, there is a space, guys. Is the space is available? No, right? What you will do if you want more space means you will fold it very nicely and you will keep, right? How many of you will fold like that? Give me thumbs up. How many of you will fold nicely and you will keep? I think Nandish. Yeah, tell me Nandish, Nagveni, Sushma, Vibha, Surya. Everyone will do that, no, guys. So we will fold the things and we will keep that, right? Because we want a large space, right? So that's right. Okay. Yes, very good. Nandish also will do. Yes. So now, first function is increasing the surface area. Second one is respiration, right? For making the ATP molecule. In the exam, if they ask, where is the respiration in the prokaryote or bacteria will take place? Someone will write that, ma'am, mitochondria. How come the mitochondria will come in prokaryotes, guys? Answer is mesosome, right, guys? Mesosome. In the mesosome, the respiration will take place. Okay. So now let us come to the replication. So it is also helps in the replication of DNA, guys. And in the reproduction also means binary fission will happen in the bacteria, right? So this mesosomes helps in the binary fission of the bacteria also. Okay. So then it will secrete some of the enzymes, and the main function, it will secrete the content which is required for the cell wall formation from the mesosome. So you know there's the proteins and some of the things are required for the forming the peptidoglycan layer, right? Cell wall. So that is also synthesized by the mesosomes only. Okay, guys, from the mesosomes. Okay. Very good, Nandish. Good idea. Okay. So then what are the functions, guys? Mesosomes, what we studied? These are the invagination of the plasma membrane, increase the surface area, and it helps in the respiration, replication of DNA, reproduction, and secretion of the enzymes, and what? Cell wall synthesis. Recall, guys. Remember. Okay. Fine. So this is about the mesosome. Let us go to the next, nucleoid. So before only we have studied that bacterial DNA is naked, right? It does not have any protein like the histone protein on it, right? So what happened in the eukaryote? DNA is there. On the DNA, we are having the histone proteins, right? So we are having the histone proteins, right, guys? But here what will happen? So the DNA is does not having the any histone protein, so it will be like this, right? It will be coiled like this, right, guys? Yes. So here you can see here it is having the DNA, right? So this we will call it as, this DNA we will call it as, guys, genomic DNA. Remember. So this DNA we will call it as genomic DNA. Genomic DNA. Which are the proteins are required, whatever the things is required, the mainly given by the genomic DNA. Okay. It is having all the it is having all the nucleotides which is required for the protein synthesis that is required for the cell. Okay, guys. The main genome, okay, that is the genomic DNA. How the nucleotide form the closed part with the help of NHC. What do you mean by NHC? Okay, guys. So that is about the genomic DNA. Okay, we know that genomic DNA is a circular double stranded. Circular double stranded. Recall, remember. Okay. So it is circular double stranded. Double stranded. Okay, guys. So now let us come to the 1 minute. Yeah. So now let us come to the see, genomic DNA, and it is a double-stranded DNA. We know that. So that is a circular DNA, right, guys? So the main enzyme, those all the things were secreted by this genomic DNA. So now let us come to the another DNA is there, right, guys? So see here, I think you have studied in the biotechnology where we will use the plasmids, right? Can anyone tell me plasmid name, guys? Plasmid name? Non-histone chromosomal protein. Yes, yes. Non-histone chromosomal protein. Yes, Nandish. PBR 322. Yes, guys, PBR 322. So we know that. So what are the main characteristics of the plasmid, right? Yes. So here you can see the bacteria is having the plasmid DNA that is mainly helping in the defense mechanism. Okay, guys. So that is mainly helping in the defense mechanism. Defense. For example, it is having the antibiotic resistance. Antibiotic resistance gene. Antibiotic resistance gene in it. Okay. So it is having the antibiotic resistant gene. So that is the plasmid. You know, some of them will think that, ma'am, is that genomic DNA and the plasmid DNA will replicate together? No, guys. In one bacteria, it may be 3, 4 or 10, 15. So many plasmids should be there, will be there. Okay. Not like only one plasmid. There will be a 2, 3, 4 plasmid will be there. Okay. But genomic DNA will be only one. Okay. Only one genomic DNA will be there. Okay. And one more thing, the genomic DNA will replicate by itself only, whereas the plasmid DNA will replicate by itself. Not related. Okay, guys? Not at all related. And from the plasmid DNA, Nourishing protein is coming for the cell purpose. Okay, without plasmid DNA also our cell will survive. Bacterial cell will survive. Means it will give the antibiotic resistance. Okay, that's it. Ma'am, this plasmid will process the central dogma. Yes, it will process wherever you can see the DNA, it will process the central dogma. Okay. Yes, because in the plasmid you can see antibiotic resistant gene. Okay. So where it will produce some of the chemicals, right? So that will be the protein, right? So first it should be converted into the mRNA, then it will secrete the protein, right, guys? Wherever you can see the DNA, it will process the central dogma of the molecular biology. Okay. Yes. So now let us come to the plasmid. I think you okay about the plasmid. So that is a double circular DNA. Okay. Double circular DNA. So we will call it as extra chromosomal. 1 minute. We will call it as extra chromosomal. Extra chromosomal. Chromosomal circular DNA. Circular double stranded DNA. Remember guys. Double stranded. Okay, in the statement wise, you will get these type of the questions. Self-replicating. Yes, it is a self-replicating. Yes, it is the self-replicating. Very good, Nandish. Self-replicating. Okay. So then this is about the plasmid. Okay, guys. So now let us talk about the cytoplasma. Okay. So now let us talk about the cytoplasma. Cytoplasma, you all know that, right? So this is like the fluid-like structure which is present inside the cell, right, guys? So wherever the other things like our nucleoid and the ribosome and this plasmid and all the things are suspended in it, right? For example, soup. If you take the soup, in the soup you can see all the veggies, right? All the veggies you can see, right? So like that, so it was all the things are suspended in it, right, guys? Yes or no? Cytoplasma, okay? So it is the area of all the metabolic function. Now you tell me, guys. So you have studied over the respiration, right? So now you tell me where the glycolysis will take place in the bacteria. Tell me. Yes, we can also call it as a cytosol. Yes. Cytosol means what? Without the organelles. Only fluid. Only fluid we will call it as a cytosol. If the organelles are present, then we will call it as cytoplasma. Remember, cytosol is only when organelles we removed, only the fluid is there. Then we will call it a cytosol. Okay, fine. Now you tell me, guys, where the glycolysis will take place. Very good, Nandesh. Very good. Cytoplasma. Yes. Okay. So all the metabolic reaction, guys, the breakdown and synthesis, you know about the anabolism, catabolism, right? So these all the things will take place in the prokaryote, that is in the cytoplasma. Okay, guys? Yes. Metabolic functions will take place in the cytoplasma. Then in the bacteria, we can't see any type of the cytoplasmic streaming. What do you mean by cytoplasmic streaming? For example, if you take amoeba. Okay, guys. In amoeba, cytoplasmic streaming is there. How it will happen, I will tell you. For example, this is the substratum. This is the amoeba. It will move like this, right? It will move like this, right, guys? Right? That is what we will call it as cytoplasmic streaming. Okay. With the help of cytoplasma, it will move. Okay. Right, guys? Cytoplasmic streaming. Okay. So we will also call it as no cyclosis. Cyclosis means cytoplasmic streaming only. Okay, guys. So now we will talk about extracellular structure. What are the extracellular structures we will see in the bacteria? So here you can see the tail, very long tail you are observing. Right. What is the tail we will call it as, guys? So here I have a question for you. Now you tell me, it is a flagella. We all know that that is flagella, right? Easy question, ma'am. Why you are asking this? Yes. Now you tell me, guys, this flagella is made up of what? Can you tell me, guys, if you know me? Flagella is made up of what? Question for you. Yes, Lakan, very good. Lakan, can you tell me, it is made up of what? Flagella is made up of what in the prokaryotic cell? Tell me fast, guys. Flagella is made up of what? Very easy question. Some of them will think that, ma'am, microtubule, right? No, guys, no. Whenever you see the flagella in the prokaryote, that is different. No, not a glycocalyx. Glycocalyx is like an outer structure, that's it. Flagella is a different. Glycocalyx does not have any like the mechanical function and those like kind. Okay. In the bacteria, the flagella is made up of flagellin protein. Remember, guys, flagellin proteins. Okay. Flagellin protein. Okay. So when you are talking about the flagella, it is made up of the flagellin protein. That's it. Okay. We'll talk about the flagella in detail. And let us see about the pillars and the fimbriae. Okay, guys. What is fimbriae? Here you can see the outer structure. It is extending, right? This all the like, right? There is a spiny-like appearance you can see, right? Spiny-like appearance. Are you observing, right? So that is nothing but fimbriae. Okay. So what is the function of fimbriae, guys? Fimbriae is helping the attachment to the substratum. Okay. So the bacteria is there. Sorry, substratum is there. The bacteria will come and attach, right? With the help of fimbriae. Okay. So with the help of fimbriae, it is helping in the attachment. Okay, fine. So then what is the function of pillar? Do you know, guys? Yeah, brushy surface. Yes. So what is the pilus now? We know that in the prokaryote, there is no male and female is there, right? Then also sexual reproduction will take place. That is not true sexual reproduction. That is a false, right? Not a true sexual reproduction, but the pilus will form between the two bacteria, right, guys? What we will call it as sex pilus. Sex pilus. Recall sex pilus, right, guys? Very good, very good. Sex pilus, right? Yes, here you can see sex pilus will form, right? 1 minute. Yeah, we will call it as a sex pilus, guys. Okay, sex pilus, which is helping in the transferring the plasmid DNA from the one bacteria to the another bacteria, not genomic bacteria, not the genomic DNA. See here, one bacteria is there. Okay, so the another bacteria is there. Okay, so in between these two, there is a connection is there. Okay, so that connection we will call it as a sex pilus. Okay, guys, sex pilus. For the sex pilus, this pilus is going to be held pili. Okay, guys. In one bacteria, it will be up to 1 to 4 pilus will be there. Okay. In some, it will help in the attachment also. Okay. In some, it will help in the attachment also, and it is helps in the sexual reproduction also. Okay, guys. Recall, remember. Now you tell me. Pilus and fimbriae is made up of what? Pilus and fimbriae is made up of what? Tell me, guys. Pilus and fimbriae is made up of. Easy, guys. One protein is there. No, not flagellin protein. It is a pillin. Okay, guys. Pillin protein. Pillin protein. Okay, guys. It is made up of pillin protein. Okay. So, pilus and fimbriae is made up of the pillin protein, and the flagella is made up of the flagellin protein, not the microtubule. In prokaryotes, I am telling. Okay. It is made up of flagellin protein. Okay, guys? Fine. So that's why it is helps in the bacterial conjugation. Conjugation is nothing but what I have told you now. That is the sex conjugation will happen. No, that is the same thing. Okay. So do you understood this, guys? Can you tell me yes or no? If you understood means give me thumbs up or some, yeah, some different smiley you are having. If you have any doubt, you can ask me, guys. Yes. Okay. No one is telling whether you understood or not. Who is this? Yes, Nandesh, understood. Okay, fine. So let us move to the next guy. Okay. Yeah. Now let us come to the ribosome. We know that it does not contain any type of the membrane, right? So guys, now you tell me, in the membrane lacking organelles, which are those? Only ribosomes? Another one is there. Membrane lacking organelle. Tell me, guys, another one is there. Which is that? First, I think you know this. It is present only in the, I will give a clue to you. It is only present in the eukaryotes. It is not present in the prokaryotes. Tell me. And, sorry, it is not present in the plant cell. It is present in the animal cell. Now you tell me, guys. It is not, this organelle is not present in the plant. It is present only in the animal. That is also not contain the membrane. Tell me, which is that? I think people from the YouTube, you can give me the answer. Guys, answer me. Which is that? Special protein. No. Peroxisome is also having the membrane, guys. Centrosome. Okay, centrosome. Okay, the centrosome will form the microtubules, right? Spindle fiber, right, guys? Centrosome is made up of the microtubules. Centriole we will call it as. Okay, so ribosome and the centrosome lack membrane. Okay, guys. So now let us talk about. So we know that ribosome is a protein factory, right? So it will synthesize the protein, right? It is a non-membranous structure. And have you heard of the polyzomes, guys? Polyzomes. What will happen in the polysome? For the one single mRNA, for example, if you take the one single mRNA, so many ribosomes will attach together. Okay, so many ribosomes will attach together. Here you can see one mRNA is there. So many ribosomes will attach together and it will synthesize the protein. Okay. Excuse me. Okay. So here what happened? For the one mRNA, so many ribosomes will attach and synthesize the protein. In which condition it will happen means whenever that protein is required in the large amount. Okay. So on that time, this type of the polysomes will be formed, guys. Okay, guys. Okay. Polysomes we will call it as, or we will call it as a polyribosome. Polyribosome. Okay. See here, single mRNA is there. Then so many ribosomes are there. Right? So many ribosomes are there. Right. So that's why we will call it as a polyribosome. Okay. It will be present in prokaryote also, in eukaryote also. Okay, fine. Then you know that in the bacteria, the ribosome is 70S type. 70S type of the ribosome. Right, guys? So here you can see 70S. Ma'am, what is S here? Right, guys? What is S? But the polypeptide chain between the amino acid. Nandish, I didn't got what you are telling. We are talking about the membrane. Membrane lacking, Nandish. Yes. What you are telling? Yeah. It will help in the protein synthesis, right? It will form the polypeptide bond. Yes, Nandish. Yes. It will form the polypeptide bond between the two amino acids, right? Yes. Correct. So then coming to the 70S type of the ribosome is present in the prokaryotic cell. What do you mean by yes? Nothing else. Very easy. Swedberg coefficient, we will call it as, okay? Or sediment constant, okay? Sedimentation constant or the Swedberg coefficient, okay? So S means yes. So very good, Anish. Yes. 70S means what? 50S plus, not 50S plus. Just we have to write that 50 and 30S. Okay, not plus. Okay, guys. 50S and the 30S. Okay. Then so many people what they will do? They will plus. Okay. Ma'am, we know the addition it is coming 80. Why you are writing 70? That is not about the plus addition and the subtraction. Okay. This is about the sedimentation constant. Okay, what is the sedimentation in the higher class? You are going to study that. Okay, fine. So let us come to the inclusion bodies. Okay, in the bacteria we can see the inclusion bodies. What is this inclusion body? You can see in the beginning only I have told you that gas vacuoles. Okay, guys. In the gas vacuoles we can see the gas vacuoles are present. So it we can see in the cyanobacteria, purple bacteria, and in some green bacteria. So where they are living in the aquatic environment. Okay. So it will help in the buoyancy. Okay. It will help in the buoyancy. You can see the bacteria will come in the upper side, right? So in the buoyancy, on the surface. Okay. So it is helps in the buoyancy. Remember, guys, it is very, very important. It is helps in the buoyancy. Okay. What is that? Gas vacuole helps in the buoyancy. Okay. So now let us come to the here only they are written the provide the buoyancy. Let us come to the food vacuole, food reserve. Okay. So it will engulf the food. Okay, it will engulf the food. That we will call it the food vacuole. Then it will store the food. What does the food it will store? It will store the starch granules we can see, starch vacuoles we can see, inclusion bodies, and we can see the lipids and cyanophyacin granules. Okay, cyanophyacin granules and the lipid granules we can see, and the starch granules we can see. Okay, guys, fine. So other than that, there is another one is there. So that is the inclusion bodies. So that is the inorganic inclusion, or we'll call it the inorganic granules. Okay. So in that we can see two types. One is the phosphate and another one is the sulfur. Okay, guys. So what you studied in the inclusion bodies, guys? So nothing. So it will storing. Okay, it will store some of the components. Okay. So in this, the first one is the gas vacuole and the food vacuole, and third one is the inorganic vacuole. Okay. So inorganic body. Okay. In the gas vacuole, it helps in the, it is filling the gas inside it, and where it is helping in the buoyancy. Okay. And in the food vacuole, it will store the starch, lipid, and the cyanophysin. Okay. So then coming to the inorganic, it will store the phosphate and the sulfur. Okay, guys, do you understood this? No, Nandish, it won't be like it won't be diffuse. Okay, fine. So let us come to the next. Yeah. So let us talk about the vacuoles, guys. Okay. So inclusion bodies we have studied. Now let us talk about the vacuole. Whenever you see the plant cell, in the center there is a big vacuole we can see. Right, guys? Big vacuole we can see. So that is a central vacuole. So that is a very large. It is like the 90% it is cell of the plant. It is made up of the vacuole only. Okay. So now let us come to the animal cell. Animal cell, it is contain only the small vacuole, or we will call it the negligible vacuole. Okay. Okay. So in the plant, we can see the central vacuole. It is helping in the what, guys? In the osmotic potential maintenance. Okay. In the osmotic regulation, it is helps. Okay. What is a vacuole? So what it will help? For example, if you see, for example, if you take the what we'll call dry grapes. Okay, dry grapes if I take. Okay. So if I put it in the water, what will happen, guys? What will happen? It will become turgid or flaccid. Yes. Yes, Nandish. So now you tell me, guys. So when I put in the water, it will become the turgid or flaccid. It will become turgid, right? It will take the water inside it, right? Because that is dry, right? So it will take the water inside the cytoplasma. So then it will take the water from the cytoplasma to the vacuole. Okay. So in the vacuole, what will happen? So it will store all the waste materials like the water and the inorganics like the ions, calcium, potassium. So like that ions will be stored inside the vacuole. Okay, guys. So whenever the cell was in the flaccid stage, okay. So on that time, what will happen? The water from the cytoplasma will move out. Okay. So then from the vacuole also the water will move out. Okay. So likewise, it will help in the osmoregulation. So that is in the plant. Okay. So now let us come to the food vacuole. Okay. What happened in the food vacuole? It will help in the digestion and the secretion of the waste. So that is about the food vacuole. Okay. And it is engulfing in the food. So now let us come to the contractile vacuole. Contractile vacuole is very, very important. So that we can see in the amoeba and in the paramecium. Okay. In the amoeba, paramecium, we can see the contractile vacuole. So that is helping in the osmoregulation. Remember, guys, osmoregulation. It is helping in the osmoregulation. You know about what is the osmoregulation, guys? So how our kidney is working, right? We have the kidney, right? So it is helping in the osmoregulation. But what about the amoeba and the lower organism? It does not have the kidney, right? So in the bacteria, if you take the amoeba and the paramecium, it is having the contractile vacuole where it is helping in the osmoregulation, maintaining the salt and the water balance. That's it. Osmoregulation, not a very big word. Okay. So it is helping in the water and the salt balance. That's it. Okay. So it is helping in the water and salt balance. Water and salt balance. Okay. Fine. So now let us come to the whether the vacuole is membrane bound or not. It will swell up. Yes, Nandi. Yes, Navos. Very good. Now you tell me, guys, whether the vacuole is membrane bound or not. Nandish is telling no. No. Nandish is telling no. What about, guys? What about other guys? It is having membrane, guys. The vacuole is having membrane. That membrane we will call it as tonoplast. Remember. So remember one thing I am telling. In the cell organelle, there are some organelles that is having the double membrane, guys. Double membrane. Okay. If they ask in the exam, which are the cell organelle containing the double membrane, I will tell you. One is nucleus. Okay. Nucleus is having the double membrane. Mitochondria is having the double membrane. And chloroplast is having the double membrane. Okay, guys. Then if they ask about single membrane, which are the organelles is having the single membrane? Okay, which are those, guys? Which are those it is having the single membrane? If you take the peroxisome, if you take the endoplasmic reticulum, if you take the Golgi apparatus, okay, if you take the vacuole, and if you take the lysosome, right? Remember, in the prokaryotes, no membrane. In the eukaryotes have the membrane, right? It is having the membrane, but some of the organelles having the double membrane, two membrane, right? Double membrane. If they ask about the double membrane, nucleus, mitochondria, chloroplast. If they ask about the single membrane, peroxisome and endoplasmic reticulum and Golgi apparatus, vacuole and lysosome. Those are having single membrane. Okay. The plasma membrane in the vacuole, we will call it as tonoplast. We will not call it as a plasma membrane. Recall, guys, remember. Okay. We will call it as tonoplast. Okay. Tonoplast. Remember. Okay. Nandish, artha agidya? Nandish, do you understood? Now? Yes, very good, Nandish. Thank you. Fine. So that's it for today, guys. In the tomorrow's class, we will continue the other organelles. Okay. So now here is the question for you. You can answer me to this question. Not Nandish. Simply I'm asking. Simply whether all of them are understood or not, like that. Okay. So you can answer to this question. It is very easy question, right? Tell me. The question is, select the wrong statement. Okay. Option A, the bacterial cell wall is made up of peptidoglycan. Okay, guys? Fine. My God, Nandish, you can send me one time. Enough. Okay. The bacterial cell wall is made up of the peptidoglycan. And option B, the pili and fimbriae are mainly involved in motility of bacterial cells. Option C, cyanobacteria lack the flagellate cells. And option D, mycoplasma is a wall-less microorganism. Which is the wrong statement, guys? Now only we studied that. Very good, very good. Option B is the wrong, right? Right. So our flagella is helping in the motility, right? Flagella is helping in the motility. Flagella, right, guys? Flagella is helping in the motility, right? Motility means what? Movement, right? Motility. Whereas, so that's why it is the correct statement. Whereas, wrong statement. Bacterial cell wall is made up of the peptidoglycan. Yes, that is correct. Coming to the cyanobacteria lack the flagellated cell. Yes, correct. Then coming to the option D, mycoplasma is the wall-less microorganism. Yes, guys. So the bacteria which is not containing the cell wall means that is a mycoplasma. We will call it as PPLO. Plural pneumonia-like organism. Okay, guys. Fine. So that's it for today, guys. Let us meet in the tomorrow's class. In the tomorrow's class, very, very important topic I will do, guys. So please, whenever you are coming to the class, read and come. Okay, read and come. In the tomorrow, we will complete this. Okay. Yeah. Yeah, I will come with the more questions. Okay. Yeah. Take care, guys. Bye-bye. If you have any doubt, I will be here only. You can ask, guys. Try to solve more questions, okay? These are the important topics.